Okay, everyone, this is the video lecture for Chapter 12 on cell division, or what we more commonly refer to as mitosis. So the ability of organisms to produce more of their own kind is one of those key characteristics that we use to distinguish living things from non-living things. The continuity of life is based on the reproduction of cells, or what we call cell division. What I mean by continuity of life is the ability of organisms to produce more of their own kind. So for example, squirrels, when they produce more squirrels. When humans reproduce, we produce more humans. That's continuity of life, and it's based in cell division. All right, so for example, in single-celled organisms, division of one cell reproduces the entire organism. This is one way that I mean that cell division is important for reproduction. Single-celled organism like an amoeba, when it goes through the process we're going to talk about here in Chapter 12 of cell division, that's how it reproduces. I mean, it splits into two, and you have two new cells, and each single-celled organism is in itself an entire organism. All right, now, multicellular eukaryotes, such as ourselves, depend on cell division for a number of things. First of all, we depend on it for development from a fertilized egg. Okay, so embryological development is one important aspect where cell division comes into play. We depend on it for growth. So as your body grows, that's cell division. You're putting on more cells on a daily basis than you're losing. And we depend on it for repair. So tissue repair, injury and wound healing, that sort of thing. So cell division is a really important part of what we refer to as the cell cycle, all right? And we're going to be talking about the cell cycle throughout this chapter, but the cell cycle basically describes all of the events that take place in the life of a cell from the time that it's produced until it goes on to divide and make two more cells. Okay, so most cell division results in what we call daughter cells. And these daughter cells have identical genetic information, or DNA, right? And so that type of cell division is referred to as mitosis. Okay, this is the kind of cell division that produces the vast majority of cells in our body. So we're talking about muscle cells, bone cells, skin cells, that kind of thing. Mitosis produces genetically identical daughter cells. Now, that's the subject basically of Chapter 12, is mitosis, all right? In Chapter 13, we're going to talk about a different form of cell division called meiosis. And meiosis produces gametes, which are sperm and egg. And we're going to see in Chapter 13 that there are some fundamental differences between meiosis and mitosis, but basically they're very similar overall. The differences that we see between these two result in the gametes produced by meiosis being genetically different from each other. So every egg that's ever produced is different from every sperm that's produced is different from every other. All right, so before we get on to that, let's talk about DNA. And because essentially what's going to be happening here, or at least what we're going to focus on in this chapter, is what is happening with the DNA as the process of mitosis or cell division moves forward. Okay, mostly we're going to focus on the chromosomes and how they're arranged and organized and move and that type of thing. But when we're talking about all of the cell's DNA, we use the term genome. Genome refers to, nuclear DNA, that's DNA inside the nucleus. It refers to mitochondrial DNA, what we call mtDNA, and it refers to chloroplast DNA, what we call cDNA. So in plants you have nuclear DNA, mitochondrial DNA, and chloroplast DNA. In animals you just have nuclear and mitochondrial DNA. But the point is, when we talk about all those together, we're referring to what's called the genome. Now, for the most part in this chapter, we're going to be dealing with nuclear DNA, okay, and see how it's arranged, organized, and moved around during the process of cell division. Now, part of what happens to the DNA just prior to cell division and during cell division is that it becomes more organized or better organized than it is typically. Basically, it forms into discrete structures called chromosomes. So chromosomes are just basically chunks of DNA, large chunks or pieces of DNA, all right? And the reason why our DNA is formed in the chromosomes is because that's a convenient way to organize it to ensure that each of the two daughter cells that result from cell division get a complete set of chromosomes or of DNA. All right, so let's take a look at a cell that's actually beginning the process of cell division or mitosis. So what you see here is the entire cell. That's the cell membrane. And then inside here we see the nucleus. And you can see all this sort of squiggly, purplish, blue material. That is all the DNA that is in the process of being organized into distinct chromosomes. All right. Now, in most organisms, there are two copies, two complete sets of the DNA inside that nucleus. So, for example, humans have a total of 46 chromosomes. That's our chromosome number, all right? Now, 23 of those chromosomes come from your father, and the other 23 come from your mother. And they are essentially, well, not quite duplicate sets, but they contain the same information. So you have two duplicate sets of information, essentially, okay? And all that DNA fits inside that nucleus. So when you consider that one complete set of 23 chromosomes in humans, if you could take it out of the DNA, or take it out of the cell rather, and unfold it, stretch it out, would be about 7 feet in length. You have two copies of it, so you have about 14 feet of DNA all packaged inside this microscopic nucleus. All right. Now, typically you can't see that DNA until just before cell division, when this process of condensing into individual chromosomes begins. And at that point, as you see here, you can start to see that DNA. So at this point, what we call that DNA is chromatin. All right, it has not fully formed into chromosomes yet, but it's in the process of doing so. All right, so the chromatin then is a complex of DNA and protein. And so what you have are proteins called histones that act like spools. And the DNA comes in and wraps around one, runs to the next, wraps around that, runs around the next one, and so on. And so these histone proteins help to organize that DNA, help to condense it and pack it more tightly so that it can actually fit inside that nucleus. But the term chromatin that you see here refers to the DNA and those histone proteins that the DNA is spooled or wrapped around. What you're seeing here is chromatin now condensing into individual recognizable chromosomes. All right, so mitosis, which is what we're talking about here in chapter 12, produces one category of cells that we call somatic. So somatic cells are also known as body cells. They're non-reproductive cells. These are cells that build our bodies. So somatic cells are all the cells in your body except for gametes, except for sperm and egg. So somatic cells are nerve cells, muscle cells, bone cells, blood, you name it. And there are several hundred different types of somatic cells in our body. All right. Gametes, again, are reproductive cells. So they are sperm and egg. So somatic cells are produced through the process of mitosis. That's what we're talking about here in chapter 12. Gametes, chapter 13, are produced through meiosis. Okay? All right. So let's talk about chromosome structure a little bit. So just prior to cell division, as we're going to see, the DNA inside the cell is duplicated. And again, this is because we need to have two complete sets of it to distribute to the two daughter cells that we get once the parent cell has split into two. So there's a process of DNA duplication, okay? Now, Some of this terminology we're going to talk about with regard to chromosomes can be a little bit confusing because some of the terms sound similar and that sort of thing. But basically, here's how we draw, or how we would show, a chromosome prior to cell division. Okay, so we have a structure basically looks like that. What you have here is sort of this narrow pinched in area. That is called the centromere. And so all of this is DNA. DNA very tightly packed into a complex, or a compact structure called a chromosome. All right, so that is a chromosome. In particular, it is a non-replicated chromosome. That DNA has not been copied yet. Now, once that DNA is copied, we have a chromosome that will look basically like this. All right. So again, we have our centromere here in the middle, same as we did over here. But now this chromosome consists of two what are called sister chromatids. These sister chromatids are duplicate copies of one another. They are duplicate copies of the original parent chromosome that we saw over here in the non-replicated chromosome. All right, now one important point. This is still one chromosome. It's just that it's a replicated chromosome now consisting of two sister chromatids. This isn't that big of a deal in chapter 12, but in chapter 13, we're going to see that chromosome number changes. And so you have to be able to keep track of how many chromosomes you're dealing with. Here's the way you do it. One centromere equals one chromosome. So we have one centromere there, one chromosome, one chromosome. What's the difference? This is a non-replicated chromosome. This is a replicated chromosome consisting of two sister chromatids. All right? Okay. So here you see an electron microscope view of a replicated chromosome. You see the two sister chromatids, one there and one there. You can see this sort of narrow pinched in area, that's the centromere, like I said. And so we have one long piece of DNA here that's condensed into a sister chromatid. We have a duplicate copy of it condensed into another sister chromatid. But because we have one centromere, this is one chromosome, a replicated chromosome. All right, so basically what's going to happen during cell division is that this centromere will split, and this chromatid will go in one direction, and this chromatid goes in another direction. So they're going to separate from each other and move toward opposite sides of the cell. And in humans, where you would have 23 pairs of these replicated chromosomes, you'll have this happening at all 23 pairs, as we'll see. Okay, so here's the basic idea. I'm just showing in simpler form. So let's imagine we have a cell with just one chromosome. So here you see a non-replicated chromosome that represents one DNA molecule. You have the centromere right there. All right, after chromosome duplication, you have that now converted into a replicated chromosome, still just one centromere, you can kind of see it there, but now it has two sister chromatids attached to it, right? Represents two DNA molecules. Cell division, or mitosis as we call it, is going to split that centromere and distribute those sister chromatids to two different cells. Okay, it's more complicated than that, but that's, in a broad overview, what's happening. All right. So, eukaryotic cell division consists really of two parts. First part is what we call mitosis, and technically this is not cell division. Technically it's nuclear division. So it is division of the nucleus. All right. So for a while, at the end of mitosis, you still have one cell with two nuclei in it because the nuclear division part is done. What has to happen then is a process called cytokinesis, and that is where the cytoplasm actually divides. So the cytoplasm between the two nuclei in the one cell splits and separates those two nuclei into separate cells, okay? And those two cells will be genetically identical to each other. All right. So we have this thing then called the cell cycle, as I said, right? It's all the events that the cell goes through during its lifetime, from the time it's produced through one round of cell division until it divides to make two more daughter cells during the next round of cell division. We can take that cell cycle and divide it up into what's called mitotic phase, all right, and interphase. So the mitotic phase is when chromosomes are separating from each other and moving to opposite sides and all that. Interphase is all the other amount of time in the cell cycle. So when the cell is carrying out its normal functions, it's not actively going through cell division, in other words. So the cell's growing, it's copying its chromosomes, it might be preparing for cell division during interphase, but it's not actually going through cell division. All right, so let me erase this real quick. All right, so in interphase, what we have is smaller phases. So interphase is broken up basically into what's called G1 phase, S phase, and G2 phase. All right, so G1, S, and G2. So the G stands for gap. Back when I was learning this as an undergraduate, G stood for growth. Not sure why they changed it, but doesn't really matter. So very specific things happen during each of these phases, as we'll see, okay? Now, the cell grows during all three phases, but the chromosomes are duplicated only during S phase. So that's when we have our DNA being duplicated, okay, during S phase. The S stands for synthesis, DNA synthesis. All right. So in broad overview then, this is what we have in terms of the cell cycle. So let's imagine first of all that we have a cell that's just been produced. It's just come out of mitosis. We have a brand new cell, and it immediately starts off in interphase. So everything I'm highlighting here is interphase, all right? So cell starts off in what's called G1 phase, okay? After G1, it transitions into S phase. S phase is when the DNA is being copied, and from S phase it goes into G2 phase. And finally, at the end of G2 phase, which is the end of interphase, the cell enters the mitotic phase, where we have, as I said, mitosis, division of the nucleus, and cytokinesis. All right? Okay, so what's going on basically in G1 phase? Well, in G1 phase, the cell is growing. It's getting back up to normal size. Because you think about it, you had a cell at the end of, or at the beginning of the mitotic phase that was a certain size. It split in two, so these two daughter cells should be about half the size of the cell they came from. So G1, that cell begins to grow and get back up to normal size. It also starts carrying out its normal function, depending on what type of cell it is, okay? All right, in interphase, in S phase, as I said, that's when the DNA is being replicated. Now the cell is still carrying out its normal function, whatever it might be, but now it's taking on the added role of copying its DNA. And DNA synthesis is energetically very expensive, takes a lot of energy to copy DNA, and it can be relatively time consuming, especially in higher organisms that have a lot more DNA, for example. All right, once DNA synthesis is about complete, the cell transitions into G2 phase, and again, it's carrying out its normal function during G2, but you can also think of the cell now, or G2 phase rather, as sort of a prep phase for the cell entering mitosis. So many of the proteins and the other structures that have to be built or are needed for mitosis are actually built during G2 phase. This is also when you have all the organelles being duplicated. I mean, we were talking about the DNA for the most part being duplicated, but all the organelles, endoplasmic reticulum, Golgi apparatus, all that kind of thing has to be duplicated as well, and generally that's taken care of in G2. All right. Okay. Then we have the mitotic phase where division of the nucleus occurs. Once that happens, for a brief time you have a cell with two nuclei right here. Then cytokinesis splits those two nuclei into two separate cells by dividing the cytoplasm between them. And then we end up with two duplicate daughter cells. Each one then starts its own new interphase cycle. All right, so these photos that you see on the top here are not actually all that useful. They're some of the worst photos of mitosis I've ever seen put in textbooks. I don't know why they did this, but we're going to focus on the illustrations down here at the bottom. So there are certain things that happen during these various subphases of mitosis, okay? So first thing to note is that the phases of mitosis that we're going to talk about, two of which you see here, so prophase and prometaphase, these don't actually exist in nature. It's not as if, if you're watching a cell dividing under a microscope, the cell enters prophase, does certain things, and then stops for a while, picks back up again, goes into prometaphase, and so on. It is one smooth, continuous process from beginning to end. So that's what I mean by these phases not being real, sort of, tangible phases in the sense that you see a cell start and stop in between them. These are human constructs. They're artificial constructs that humans have developed to help us better compartmentalize and understand what's going on during the process of mitosis. Okay. All right. So first thing we have is a cell that's, say, at the end of G2 of interphase. All right. So what it's doing now is getting ready to go into the mitotic phase. So first thing, notice all that thinly sort of spaced chromatin in that nucleus. Okay. It's uncondensed. This is all going to have to condense into recognizable chromosomes. All right. Also notice we have two centrosomes. Now, we start off with one, but then toward the end of G2, a second one is produced. So what these are going to do is basically act like traffic cops. They are going to attach to the chromosomes via what are called spindle fibers, as we'll see in a minute, basically microtubules, and push those chromosomes around and line them up. Okay? All right. As the cell goes into prophase, we see now that the chromatin has been condensed. So this really starts in prophase and continues on through prophase. This is kind of a late prophase cell here. And now you see we have individual recognizable chromosomes, all right? Okay, we also see that one of our two centrosomes is now migrating to the other end or other pole of the cell. And you can see what's called an early mitotic spindle forming between the two. That's just a series of microtubule fibers, one of our three protein fiber types of the cytoskeleton that we talked about back in chapter 6. All right. Eventually, these fibers are going to connect to those centromeres of the chromosomes. But what has to happen first is that the nuclear membrane needs to break down. Now, one thing about this prometaphase, you can ignore that. Both of these I consider to be prophase, okay? Prometaphase is a new phase they've developed in recent years. I don't know why. It doesn't help anything. It doesn't clarify anything. So for our purposes, you can ignore this prometaphase and just think of this as a late prophase cell, all right? This is maybe a mid-prophase cell. So notice here in late prophase, the nuclear membrane is breaking down, disintegrating, because it's in the way. It is separating the fibers from the chromosomes. And now you can see that the fibers are attaching to the centromeres of the chromosomes. All right. In particular, if you look here, there are basically the centromere has on either side of it two protein plaques, so one there and one there. Those are called kinetochores. All right. They are attachment points for the microtubule fibers, and you see them right there. Okay, so a microtubule fiber attaches to each of the two kinetochores on either side of the centromere. Okay. All right. So that's late prophase right there. Okay. Next phase, metaphase. Metaphase is always very easy to recognize under a microscope because the chromosomes appear to be sort of lined up in the center of the cell, like you see right there. All right, so at this point, we have a machine basically called the spindle. It consists of the centrosome on one side, the microtubule fibers extending from that to the chromosomes in the middle, the microtubules extending from them to the centromere on the other side. All right, all of that is referred to as the mitotic spindle. Okay, that's the machine that's going to distribute these chromosomes to opposite sides of the cell. Okay. Now, these chromosomes are lined up in basically what is sort of an imaginary two-dimensional plane, like a plate in the middle of the cell. It's called a metaphase plate. It's not a real structure, it's just the two-dimensional plane that they line up on. All right, we'll see more about that a little bit later on. Now, once metaphase is complete, anaphase begins. And the way you know an anaphase cell is that the chromosomes start to move away from each other. So what happens is that the centromere here that's intact splits because the fibers on either side retract. And as they begin to pull, it causes the centromere to split, and the two chromatids that were attached to that same centromere now move away from each other. At this point, we actually no longer refer to them as chromatids. They are now referred to as chromosomes. They are full-fledged chromosomes in their own right. But anaphase is easy to recognize under a scope because you see space now between two distinct sets of chromosomes. All right. Anaphase ends when the chromosomes have moved as far as they are going to and they stop. Okay? All right. Now, telophase, our fourth and final phase, begins when the chromosomes in anaphase stop moving. And main things that happen here is that a new nuclear membrane begins to form now around each set of chromosomes, forming two nuclei. So for a while you have one cell with two nuclei in it. And then, a little bit later on, cytokinesis actually begins. And what you see here via what's called this cleavage furrow is the way that cytokinesis happens in animal cells. Basically, there's a ring of contractile proteins that runs around the inside of the membrane here, and it cinches down like a drawstring in a garbage bag. And so you get this trench or trough that runs around the outside of the cell called a cleavage furrow that gets deeper and deeper until eventually it sort of collects from all points at one point in the center, and the two cells separate from each other. All right, so we're going to see some of that in more detail here in just a little bit. But let's now put this in context with the overall cell cycle. Okay, so again, everything we're talking about with regard to mitosis itself, all of this is happening right here in the mitotic phase. So that may be just 5 or 10% of the overall amount of time in the cell cycle. Everything else is interphase, so G1, S, and G2. All right, so let's take a little bit closer look at that mitotic spindle, like I said, which is the machine that's essentially causing the centromeres to split, to pull the centromeres apart so that chromatids separate and move to opposite sides of the cell, okay? So let's imagine we have a cell that's in metaphase of mitosis, okay? All right, we have a centrosome on one side, centrosome on the other, and we have our fibers connecting to the kinetochores of each of those centromeres, all right? We also have short fibers on the opposite side of each centrosome, and those are what's called the aster. The point about the aster is that it sort of connects the centrosome to the inside of the cell membrane. So your cell membrane is running here and then also around there, okay? So the aster then connects the centrosome to the inside of the cell membrane. That's important because if the centrosomes weren't connected in place, when they began to retract their microtubule fibers to pull chromosomes to opposite sides of the cell, what might happen is that the centrosomes themselves would get pulled in toward the center if they weren't anchored in place, rather than the chromosomes or chromatids being pulled away from each other. The asters lock those centrosomes in place, so if anything's going to give, it's going to be the centromeres, and that will cause the chromatids to move in opposite directions away from each other. Okay, so the whole structure, let me change color here. The entire structure that we're talking about here, from one aster all the way across to the other, that is the mitotic spindle, okay? So it consists of both asters both centrosomes that you see here, okay, that one and that one, the microtubule fibers, and even the chromosomes in the middle. All of that is the mitotic spindle. That's the machine. All right. And also, don't forget, very important thing, when you look at a single chromosome, say that's a replicated chromosome like I showed you before, right? What we have is the centromere in the middle, that's here. Oops. Get my pointer to work. That's here. And then either side, you have that protein plaque, again, called the kinetochore. All right, and that's where our fibers attach, okay? Very important. All right. So let's look at a nice picture here showing a cell that's in metaphase to kind of tie all this together. Okay, so first of all, like I said, we have a cell in metaphase. You can tell that because chromosomes are sort of lined up in the middle. Now you see it, since they've sort of tipped that cell toward you, angled it toward you, you can see the chromosomes actually aren't in a single file line. They're just splayed over this two-dimensional region here called the metaphase plate, okay? If you tilted that cell back away from you, it would look like the chromosomes are lined up in single file, but they're actually not. All right, we have one aster here, centrosome, microtubule fibers, chromosomes here, microtubule fibers, another centrosome, and another aster there. Again, that whole thing is called the spindle apparatus, okay? Now, one important point. These microtubules that you see actually connected to the kinetochores, those are called kinetochore microtubules, all right? This is a blow-up electron microscope view of a single centromere. You can see one kinetochore there with the fibers extending off, and same thing here, fibers extending off. Okay. Also notice though, for example, here, and over here, and over here, and right there, you have spindle fibers that actually don't connect to kinetochores. Those are called non-kinetochore microtubules. They overlap with each other. So they grow out from this direction, and when they overlap, they bond to each other. They lock in place. The reason why that's important is because those fibers continue to grow out from their respective centrosome, but now here, where they're locked together, they can't slide past each other anymore. So because those fibers are still getting longer, that has the effect of pushing the two centrosomes away from each other, helping to elongate the cell. That is one of two forces that helps to sort of pull those chromatids apart from each other to split those centromeres. The other force is the retracting of these microtubule fibers, the kinetochore microtubules that retract. So they're retracting, that's helping to pull chromatids apart, but the cell is also elongating as the centrosomes here and here move away from each other because of the action of these non-kinetochore overlapping microtubules. Okay, down here you see in a light microscope view, you can see a centrosome there, one there, aster fibers here, microtubule fibers here, chromosomes lined up at the metaphase plate. That's a metaphase cell. All right, next thing is anaphase. Now in anaphase, basically what's happening is that for the most part, these microtubule fibers are shortening. And there's an enzyme that causes those proteins to be cleaved, which makes, or cut rather, which makes those fibers shorten. It's not that important. The important point is that sister chromatids separate and move along the kinetochore microtubules toward opposite poles of the cell, okay? All right, and we talked about the non-kinetochore microtubules again, helping to carry that out by elongating the cell and making the cell more oblong or egg-shaped, if you want to think of it that way, rather than sort of spherical as shown in this illustration here. Now, cytokinesis, which we talked about happening in telophase, can actually begin in some cases earlier in anaphase and then continue on into telophase. Not always, but sometimes it does. So it's a little bit more variable than just straight anaphase and telophase in terms of when it happens. All right. So I believe I'm at about the half-hour point on this. So again, for file size limitations, I'll have to keep these at about a half hour. So we are going to stop here, and then I'll pick up in the second part of this with talking about cytokinesis. Then we'll put it all back together for the end of this chapter. All right?